hope you're all doing really well. Thank you for joining us for the next instalment of our microbiome webinar series. Today we would like to welcome Dr. Nissan Yissachar, Principal Investigator at the Goodman Faculty of Life Sciences at the Bar-ilan University. Facilitated by the Whitley A35 workstation, his research focuses on host-microbiome interactions and their impact on immune system function in health and disease. His talk is titled Thinking Outside the Mouse, ex-vivo dissections of host microbiome crosstalks. At the end of the webinar, you'll have the opportunity to have your say in our question and answer session. So please type your questions into the chat box and we will answer them at the end. On behalf of Don Whitley Scientific, we hope you enjoy the webinar.
Okay, so hi everyone, thanks for joining in. And I'd like to start by thanking the team at Don Whitley Scientific for inviting me to participate in this fantastic microbiome webinar series. Thank you very much. And let's start. So as you probably know, tissues in our body that are exposed to the external environment are heavily colonized by microorganisms, which we collectively call the microbiome. And that includes the lungs, the skin, the urogenital tracts, and of course, the gut. And there's an increasing microbial load going all the way from the stomach through the small intestine to the large intestine that contains about 10 to the 11 bacterial cells per gram of colonic content. And in a healthy individual under homeostatic conditions, host microbiome interactions can be defined as mutualistic, right? The gut microbes gets to live in a protected and nutrient-rich environment. And in return, they supply us a long list of benefits. They enhance our digestive efficiency, so we can now extract much more energy from the food we eat. They have very important metabolic functions. For example, they produce essential vitamins like vitamin K and important metabolites and short-chain fatty acids. They even protect us from pathogenic infections simply by occupying this niche and creating a competitive environment for opportunistic pathogens. And last but not least, these microbes are crucial for healthy maturation, development, and function of our immune system in the gut and systemically. And we know that, for example, when we look at by looking at the germ-free mice that are completely devoid of bacteria, we can see that the immune system of these mice is immature. There are many immunological defects, cellular changes in cellular numbers, and the disrupted proportions of immunological subsets, and many other defects. Most of these defects can be corrected simply by recolonizing these mice, adding back bacteria to these cages, but not all of these defects. And what's maybe very striking is that we understand now that specific microbial species within the human microbiome can activate specific arms of our immune system, which is, I think, really mind-blowing. So given these close connections between the gut microbiome and our immune system, it might be not surprising that changes to microbiome composition are associated with a wide array of diseases, locally in the gut, like inflammatory bowel disease and irritable bowel syndrome, but also systemically, systemic multifactorial diseases like diabetes, metabolic syndrome, multiple sclerosis, Parkinson's, are all associated with dysbiotic, not normal or unhealthy changes to gut microbiome composition. So there's a lot of research going on trying to figure out how can we harness some of these microbes therapeutically. Basically, can we supplement specific microbes to a specific individual in order to ameliorate or treat specific disease? And this is exactly what we're doing in the lab. We're trying to figure out, first of all, how does it work, right? What are the molecular mechanisms that mediate a healthy and pathologic host microbiome interactions? And second, we're trying to figure out how do changes to microbiome composition will functionally affect the gut, specifically the immune system, the nervous system, and other systems within the gut. How do we do that? Like many other labs, we use several in vitro and in vivo methodologies, but in addition, we also use another tool, which I developed during my postdoc with Diana Mattis and Christophe Benoit at Harvard Medical School a few years ago. And this is the 3D gut organ culture system. And the idea is pretty simple. We basically dissect out large intact fragments of the mouse intestine, connect it to this gut organ culture device that we fabricate in the lab. And this device contains 6 individual channels. Each channel has 2 inputs and 2 outputs connected to a single intestinal fragment. The first input is connected to the gut lumen, so we can now infuse bacteria, metabolites, drugs, you name it, directly to that specific part of the gut with precise dosing and timing. And the other input flows in a sterile, serum-free culture medium that supports tissue viability in culture. This device is connected to this set of syringe pumps that control the luminal flow and the extraintestinal flow for each one of these tissues, so we can now control intestinal conditions in each one of these cultures in real time throughout culture. When we look at these organ cultures, we can see that under these conditions, they are alive and well. We can detect gut motility and kind of peristalsis contractions in both large and small intestinal cultures over time. And we show that these conditions can maintain tissue viability for about 24 hours. Now, using this approach, we recently found that new mechanisms by which immunomodulatory gut bacteria basically exert their effects. And we showed that bacteria that induce tolerogenic responses, regulatory T cells development, Tregs, or bacteria that induce effector T cell responses like Th17 development, when they colonize the gut, one of the first responders are actually the enteric neurons, the enteric nervous system. And we found that enteric neurons sense the microbiome and respond to the microbiome. And we showed that Treg or Th17 inducing bacteria differentially activate these neurons and control the production of a specific set of neuropeptides that are produced by enteric neurons and then affect the intestinal immune system and direct it to differentiate towards inflammatory or anti-inflammatory conditions. Now, since then, we further developed this system, both technically, but also we can now extract much more information from this system and this approach. We showed that this system is a valuable tool for studying immediate early responses to specific bacterial species, to large communities of bacteria, like whole microbiome samples, to bacterial metabolites, drugs, and so on. And we investigate these connections using this system in healthy developmental stages like host-microbiome interactions at birth. And we also look at pathologic conditions, like we study leaky gut dynamics and others. Now, in this talk, I want to share with you an interesting story about how changes to human microbiome following diet will affect microbiome composition and gut responses and the underlying mechanisms. Now, over the past few years, determining microbiome composition became relatively simple or relatively accessible. We can simply collect fecal samples from patients before and after treatment, sequence these samples using next generation sequencing, and then determine basically which bacteria are there and in which quantities. The more difficult question to answer is, what does it mean, right? What are the functional consequences of these changes to microbiome composition? And can we identify causative connections between specific bacterial taxa to a specific set of genes or functions within the gut tissue? So how can we move from these long grocery lists that we get from sequencing to actually functional insights in the gut? To address this question, Hadar, Ayelet, and Efrat in my lab, together with Ehud and Steve from Kaplan Medical Center, looked at diet host microbiota crosstalks in irritable bowel syndrome. Irritable bowel syndrome, or IBS, is a functional disorder of the GI tract. It causes pain and changes in bowel habits and bloating and others. The cause for IBS is unknown, but we know that it involves different systems within the gut, like the muscular system, the enteric nervous system, the immune system, and of course, changes to microbiome composition are associated with irritable bowel syndrome. Now, there's no therapy for IBS, there's no cure, but there's a relatively new diet called low FODMAP diet, fermentable oligo-D and monosaccharides and polyols, that was developed in Australia, and it seems that many of IBS patients positively respond to this diet. Now, this diet is not a long-term solution because of its very strict diet, and it has some nutritional deficiencies. It's very difficult to adhere to this diet. So what we try to ask in this project, basically, we know that diet is one of the major factors that affect microbiome composition. So we asked in this project, what are the mechanisms by which low FODMAP diet positively affect IBS patients and ameliorate disease symptoms? And the hypothesis was that maybe low FODMAP diet will change microbiome composition, which later on positively impact intestinal functions, maybe neurological, maybe immunological, and eventually will improve and ameliorate IBS symptoms. How do we address this question? We started with establishing a small clinical cohort of newly diagnosed IBS patients and therapy-naive IBS patients. We collected fecal samples before, during, and after these 6 weeks of low FODMAP diet, and we sequenced them. And using 16S sequencing, we found indeed interesting changes to microbiome composition. We showed that clinically about 7 out of the 10 patients reported improved in their symptoms. And we show that over time, over this 6 weeks period, we can detect specific changes in microbiome composition. We can see that some bacteria, the abundance of some bacteria was increased, and some bacteria, their abundance was decreased on this diet. Now, again, we're going back. This is interesting, but I know it's not new. So the more difficult question to answer, as I said, is what does it mean, right? How do these changes to microbiome composition will functionally affect the gut? To address this question, we used the gut organ culture system, and we infused into these gut fragments whole microbiome samples collected from the same patient before and after low FODMAP diet. And after a short period of culture, we dissected early intestinal transcriptional responses by RNA sequencing. And when we did that, we found, very interestingly, that post-diet microbiome will differentially affect tissue transcription. It will post-diet microbiota induces neuronal function and neuromuscular functions and the pathways associated with the extracellular matrix and cellular adhesion, while suppressing inflammatory effector immune responses like TNF and IL-1 and other TLR signaling and so on. So that was the response to whole microbiome community. Can we identify specific microbes that are that mediate these responses? So here we took advantage of another advantage of the gut organ culture system. We know exactly which microbes we infused into these tissues because we sequenced them, and we know exactly what was the intestinal response because we sequenced that as well. So we can now correlate bacterial abundance with cellular responses. And when we did that computationally, we predicted that a specific bacteria, Bifidobacterium adolescentis, seems to be significantly correlated with a set of intestinal genes. What is Bifidobacterium adolescentis? This is a Th17-inducing bacteria, right? When we transfer this bacteria, this human symbiont, when we transfer it to germ-free mice, it induces effector T helper 17 responses. That was shown a few years ago. And in our cohort, the abundance, the intestinal abundance of Bifidobacterium adolescentis was diminished in response to low FODMAP diet in almost all patients. Now, which genes were correlated with the abundance of B. adolescentis? When we look, we identify immunological genes and genes associated with cytoskeleton and cellular adhesion. We also know that this bacteria was shown to be able to adhere to the epithelial barrier, the intestinal epithelial barrier, which is critical to maintain intestinal homeostasis. These connections between these tight junction connections between epithelial cells in the gut are crucial to maintain gut homeostasis. And when they are disrupted, this is a kind of a leaky gut condition, they promote inflammation and disruptions to intestinal function. So given these evidences, we hypothesized that maybe Bifidobacterium adolescentis alters gut barrier functions in IBS and triggers a leaky gut, which is one of the hallmarks of irritable bowel syndrome. How do we address this hypothesis? We use the Don Whitley scientific culturing system to culture Bifidobacterium adolescentis, which we predicted to be a barrier-disrupting microbe, or B. fragilis as a kind of a control bacteria that should not alter the barrier. We co-culture these bacteria in vitro with the intestinal epithelial cell line, CACO2, and then we looked at the tight junctions structure and the epithelial barrier integrity. When we look at one of the major tight junction proteins, ZOO1, we can see that addition of Bifidobacterium adolescentis diminish the expression of ZOO1 tight junction in epithelial cell lines, but not the addition of B. fragilis or the control cells. When we measure the intestinal barrier integrity by transepithelial electrical resistance assay or TEER assay, we can see that the addition of Bifidobacterium adolescentis, but not B. fragilis, rapidly disrupted the barrier integrity. We see that, again, the live bacteria, interestingly, the addition of live bacteria, but not bacterial supernatant, disrupts the barrier. And when we add heat-killed bacteria, we get some kind of an intermediate response, intermediate disruption to the epithelial barrier, which might suggest that this effect is mediated by maybe structural bacterial components. These experiments were performed using bacteria that were purchased from international bacterial banks, but we also, to further investigate it, we also isolated bacterial isolates, B. adolescentis isolates, again using Don Whitley scientific culturing system, anaerobic chamber. We isolated B. adolescentis strains from these IBS patients before and after diet. And we repeated these experiments when we could, again, identify and see that the addition of Bifidobacterium adolescentis from these IBS patients disrupts intestinal epithelial integrity and permeability. When we look in vivo in mice, we can see that gavaging or supplementing mice with B. adolescentis disrupt epithelial barrier in vivo, and we can see that when we measure intestinal permeability in vivo using a FITC-dextran assay, but not the gavage of B. fragilis, for example. And we can look at these, when we look at these tissues of these mice, we can see again disruption to the expression of tight junction proteins as ZOO1. When we zoom in using electron microscopy, we can see that in the small intestine, we can nicely detect the epithelial, the intestinal villi and the brush borders of these epithelial cells in untreated mice or in B. fragilis gavaged mice. But when we look at mice that were gavaged with B. adolescentis, we can see the accumulation of this strange subset, which we speculate that this is a bacterial-derived exopolysaccharide produced by B. adolescentis, and this substance coats the brush border of the intestinal epithelium. It might be responsible to the changes in the gut permeability that we detect in vivo and in vitro. We're not sure yet. What causes the changes in B. adolescentis abundance in response to diet? To address that, we followed B. adolescentis growth in vitro in different conditions. We see that we can detect, again, using Don Whitley scientific anaerobic chamber, we can detect very pronounced bacterial growth under culture conditions using rich medium. There is no growth when we use very minimal medium that contains no sugars or proteins. And then we added back a few sugars that are devoided during low FODMAP diet, for example, inulin or fructose. And we can see that in response to fructose, that is abundance in fruits, but not inulin, that is a close relative of fructose, but is abundance in vegetables, we can see that in response to fructose, we can detect bacterial growth. So we can conclude that it seems that fructose avoidance under this low FODMAP diet might be the one that's responsible to the reduced B. adolescentis levels that we detect in our patients. Now, lastly, we repeated these experiments using the gut organ culture system. We infused B. adolescentis or B. fragilis directly into the gut organ cultures. And here again, when we look at tight junction structure, we can detect this reduced expression of ZOO1 in the intestinal epithelium, and we can detect transcriptional changes that are related to the intestinal epithelial function. And the next question was, can we measure changes to gut permeability using the gut organ culture system? And to do so, Hadar and Ariel in my lab developed this ex-vivo intestinal permeability assay using the gut organ culture system. The idea is pretty simple. We infused a fluorescence substance, FITC-dextran, into the gut lumen in culture. together with the desired stimulation. And then this substance slowly diffuses out to the extraintestinal medium, and you can measure the fluorescence intensity and quantify the permeability of the gut. The higher the fluorescence, the more permeable is the gut. We can do so in our using the gut organ culture device for up to 6 intestinal fragments in parallel. And the cool thing is that since we're working on the bench, we can now do that and track changes to gut permeability in real time. So just to show you a few results, we can see that infusion of B. adolescentis into gut organ cultures results in very rapid and very significant increase in gut permeability, within the first few hours, but not control bacteria. And when we couple this assay with live imaging, we can detect changes to gut permeability in real time at the whole tissue level. What you see here are 3 fragments of colonic tissues in culture, infused with B. adolescentis, B. fragilis, or just sterile medium as a control. And using a custom-made software that we developed in the lab, we can now quantify the extraintestinal accumulation of phytidextran and track it over time. So we can now generate these single colon traces that quantify gut changes to gut permeability in response to changes to gut microbiome at the whole tissue level in real time. Okay, so just to summarize, we show that the gut organ culture system is a really unique pathway discovery platform. It takes us all the way from clinical microbiome samples to functional impacts and molecular mechanisms that connect specific bacteria to specific intestinal functions. We found that low FODMAP diet alters microbiome composition and reduces the level of Bifidobacterium adolescentis. We also show that post-diet microbiome rapidly shifts colonic gene expression, especially we see induction of neuromuscular programs and suppression of pro-inflammatory programs. We also showed that using this system, we can predict and then experimentally validate functional microbe-gene interactions. We found that B. adolescentis is a diet-sensitive and barrier-disrupting pathobiont in irritable bowel syndrome. And just before I'll finish, I just want to mention that this approach works very well in other conditions, not restricted only to the gut, but also in multiple sclerosis, for example, or in Parkinson's disease. We can take the same approach and identify new mechanisms that underlie host microbiome interactions. And last but not least, we continue to investigate host microbiome interactions using the system. If you are interested in learning more about our recent development, you can look online in this preprint in which we recently published and showed that how can we utilize this system to investigate the neuronal or neuroimmune interactions that takes place in the gut and how these are affected by the gut microbiome. And with this, I would like to end and to thank members of my lab, the funding agencies, and of course, again, the team of Don Whitley Scientific for their kind invitation. And thank you very much for your attention.
So, hello everyone, and thank you very much for attending our webinar today. And also, of course, thank you to Dr. Yissachar for such an excellent presentation.
Thank you very much, Andrew.
We do have a few questions for you that have come up as we've gone along. So, we've just had one pop up on the screen in front of me, which is a nice, easy one. What diet do you suggest for IBS patients? But that's kind of a broad question, but I don't know if you have a contribution there.
Yeah, so it's a good question. I'm not a clinician. I'm a biologist, a PhD. So I can't, if I'm going to recommend something and then I can't give kind of clinical recommendation. I can say, but that our research show that it seems that Bifidobacterium adolescentis is one of the mediators of abnormalities or changes to the gut and to the microbiome in IBS. And it seems that the low FODMAP diet, which is in clinical use and is highly recommended for IBS patients, it seems that this diet can reduces the level of that bacteria, and that seems to be correlated with the beneficial effect of the diet. Since we show that this bacteria can benefit and can utilize fructose to propagate, might be interesting to avoid, I would suggest, for example, to avoid fructose in kind of a setting of a clinical trial, to avoid fructose and not like the whole FODMAP, fermentable oligo D monosaccharides and so on. So just instead of avoiding this very large group of foods, try to avoid only fructose, which is enriched in fruits, of course, and honey. Maybe this would help some patients. There are some reports about a low fructose diet in some conditions, but I'm not sure if it is well established in IBS. But the results suggest that it might worth a shot.
Yeah, of course. Thank you. And that reminds me of a question I had myself, actually looking at your data for fructose compared with inulin, which doesn't promote growth of the organism. What about lactose? Have you done anything with lactose?
No, we haven't. But lactose is indeed one of the sugars that is avoided under low FODMAP diet. And it seems to be irritating for some IBS patients as well.
Yeah, and of course, there could be a different mechanism for that. It may be nothing to do with this organism. Yeah.
Exactly.
So I've got another one here that's just come up before I have any more of my own. Did you find a correlation between oral microbes and gut microbes in your studies?
It's a good question. We haven't looked at that. There are some reports showing correlation between oral and gut microbes, especially in the context of inflammatory bowel diseases, IBD. There are some beautiful work by Kenya Honda in Japan showing that the translocation of oral bacteria to the gut can initiate inflammation and inflammatory bowel disease in a mouse model. But we haven't looked at that in this setting yet.
Sure, thank you very much. So here we have, can the gut culture system be used for maintaining other intestinal tissues, for example, human gut tissue?
Right. So I get this question all the time. It's theoretically, I guess it could work. It of course will be required some serious adaptations to the culture setting. The human intestine is much larger than the mouse one. In general, for organ cultures, the larger the tissue, the more difficult it is to keep it alive, to keep it viable under ex-vivo conditions. My kind of gut feeling is that once we can adapt the medium, the supplements, the culture conditions in a way that it will support the culture of human gut for, I guess, at least a couple of hours, which will enable us to perform some experiments at that time frame. We are still working on that. It will require some also accessibility to these tissues, usually after surgery. And but what we're doing now is basically, it's much easier to obtain tissues from mice. So we use human microbiome samples and introduce them to mice tissues. So it's kind of a hybrid system.
Yeah, that makes sense. Thank you very much. So touching on something we briefly discussed earlier when I asked about lactose. So in addition to IBS, does Bifidobacterium adolescentis get involved in any other pathologies? Because I thought this was generally a beneficial organism.
Right. So Bifidobacterium adolescentis is, of course, a member of the Bifidobacterium genus, and it's considered to be a beneficial bacteria. There are many reports showing that this bacteria is beneficial in different disease models, like some inflammation, some intestinal inflammation in some mice models and others. And in general, Bifidobacterium considered to be kind of a good bacteria. There are some other studies showing, there was a really beautiful Cell paper by Peter Tarnbach at UCSD, published, I think, 3 or 4 years ago in Cell. And he showed that if you the transition to a ketogenic diet reduces the level of bifidobacterium in general and bifidobacterium adolescentis specifically, and results in the reduction in T helper 17 cells in the gut, which are important for effector immunity and for protections from infection and others. So that was shown in that context. It was also reported that B. adolescentis is sensitive in some other diets, and there are some preliminary data in other models of autoimmunity. But yeah, I think one idea of how to solve these discrepancies, it was shown a few years ago by, I think, Eric Paymer. He showed that different strains of Rubinococcus gnavus, which is considered to be a pathogen in the context of inflammatory bowel disease, he showed there are different strains of the same bacteria. Some of them can act or can produce the toxin and can function as a pathogen, and some of them will not. So I guess that might suggest that maybe even for B. adolescentis, we might actually look at different strains in different conditions and different microbiomes in different humans. And I guess under different environmental conditions or different pathologies, different strains of B. adolescentis will come into play and will maybe differentially affect the gut and the functions of the gut.
Yeah, it's always a complex interaction, isn't it, within the microbial community? And someone's just asked about that. How does the coexistence of microbes have an effect here?
Yeah, it's a good question. It's very difficult to address. Definitely, the B. adolescentis or any bacteria are not living by themselves in the GI tract. It's always part of a very dense ecological niche. And again, I think we just don't know enough. We don't know the details yet, but B. adolescentis, as many other bacteria, will definitely be affected by the presence of other bacteria in the gut. And my guess, it will affect its function and its ability to increase gut permeability in that case.
Yeah, of course. And so we've talked a lot about B. adolescentis. You touched on Rubinococcus gnavus there. And so what other bacterial species might have a role in IBS, do you think?
So that requires very high resolution and characterization of the gut microbiome and in much larger cohort. That was performed, and one example is a Cell paper published at 2020 by a group that characterized microbiome composition and metabolomic composition in IBS patients in a very large cohort in a very high resolution. So, for example, they and others found that the proteobacteria, for example, are increased in IBS. Other groups also talked about Coprococcus, Ruminococcus, and other types of bacteria that actually these groups of bacteria are considered to be troublemakers in other diseases as well. But in that Cell paper that I mentioned before, at 2020, they also characterized the metabolome of IBS patients, and they found connections between the metabolic activity of the microbiome to the flare-ups of the IBS. Yeah. So that was also very interesting as well.
Okay, thank you. I'm just scanning my screen for any further questions, but I think that is all of them for now. So unless we get contacted afterwards, of course, which does happen, and I'll relay them to you then. But for now, I'll just say thank you very much, Nissan, and thank you very much for everyone attending today. Thank you.
Thank you very much.
Don Whitley Scientific has thousands of satisfied users of our anaerobic and hypoxic workstations around the world. We are now proud to launch the latest addition to the range, the Whitley A25 workstation, which is the most compact model in our range. The A25 is equipped with many unique features to ensure easy and efficient use and to guarantee that the very best anaerobic conditions are maintained. It has a capacity of over 200 Petri dishes, while still leaving more than adequate space to process samples in a strictly controlled anaerobic environment. The Whitley Instant Access porthole system allows entry into the chamber in seconds and needs neither gloves nor sleeves. Importantly, this system also uses 8 times less gas than conventional sleeves. The built-in rapid airlock, which can accommodate 20 Petri dishes or 3 500ml Durand bottles, allows items to be transferred into the workstation atmosphere in less than 20 seconds. Doors are interlocked and cannot be opened simultaneously. Front-loading and side-loading letterbox entry systems are also available, providing a straightforward way to quickly introduce individual samples into the workstation. The A25 is fitted with a touchscreen, providing the user with an intuitive interface to control and operate the workstation. Previously unavailable on a workstation of this size, the Whitley Anaerobic Conditions Monitor and patented Catalyst monitoring system provide additional assurance of the correct incubation conditions. These systems alert users to the condition of the anaerobic atmosphere and monitor the health of the anaerobic catalyst. Whitley workstations are the only workstations to provide automatic early warnings about the anaerobic atmosphere and catalyst function. Temperature can be precisely controlled in increments of 0.1 degrees Celsius. Maintenance-free automatic dehumidification is also fitted as standard. Relative humidity can be controlled up to 80%. With a wide selection of options available, the A25 can fulfill the needs of any laboratory requiring a compact anaerobic workstation. A versatile system, the A25 can also be used as a microaerobic workstation by simply connecting the relevant premixed gas. The A25 is the ideal workstation for labs with limited space that wish to improve anaerobe isolation and reduce costs in comparison with anaerobic jars.